Dr. Samar, should we start? And Dr.
Yes,
Bergmann?
Yes, we can start now.
All right. Bismillahirrahmanirrahim. Assalamu alaikum and good day, ladies and gentlemen. I'm Hazima Mozam and I welcome you all to another exciting lecture under COMSTECH NTD joint lecture series. This is the 7th lecture in our series of lecture, and we are very pleased and delighted and honored to have Professor Dr. Bartira Bergmann with us. Professor Rosie Bergmann is from Universidade Federal do Rio de Janeiro, Brazil, and she specializes in nano and micro technologies for the anti-leishmanial drugs. The title of our talk today is New Anti-Leishmanial Drugs from Discovery to Delivery. To brief you about her in brevity, Professor Bartira obtained her BSc in biomedicine from University of Sao Paulo, Brazil, and MSc in medical parasitology from LSHTM, University of London, and PhD in biology from Brunel University, UK, on chemotherapy and immunology of leishmaniasis and African trypanosomiasis. She went on to do her postdoc from University of Toronto, Canada, on tularogenic vaccination in transplantation, later applied with success to leishmaniasis. Presently, she holds a professorship post in UFRJ, where she is a member of Animal Ethics Committee and head of leishmaniasis group working on mucosal vaccines and drug release systems in leishmaniasis. In the industry area, Bartira has developed projects with pharmas like Sanofi and GSK and founded Leishio Nano startup for the nano and the micro technologies development of novel vaccines and drugs. With this, we are very honored and pleased to have Professor Dr. Bergmann with us. And with this, I'd invite her to formally start with her lecture for today. Over to you, Doctor.
Thank you very much, Kazima, for your nice presentation. So I will share my slides. Excuse me. Can you see the presentation? Is that fine?
Yes, we can see your slides.
Yeah, and my pointer too. Can you see it moving? Yes.
Yes.
Ah, okay, great. Okay. Good morning, or good afternoon, good evening. It's very nice to be back to Pakistan. Although it's from a long distance, but it's very nice to see that there are so many students here, and I remember it was so nice to meet them in 3 years ago. No? It's a long time now. So my talk today will be on the new anti-leishmanial drugs from discovery to delivery. This sounds a bit ambitious title, and actually it is a bit ambitious because it's very wide and broad. But I try to concentrate on what we've been doing in the last years, especially the work that we carried out with the NTD group. So anti-leishmanial drugs, they are actually very old and not new at all. They are all repositioned from other diseases. And we have the heavy metal pentavalent and trivalent antimonials that were discovered in the beginning of last century. And it was first discovered by the Brazilian physician Gaspar Viana in Brazil in the treatment of cutaneous leishmaniasis, and it's still used today in the treatment of cutaneous and visceral leishmaniasis. In the 40s, we had pentamidine used for diabetes, and then the antibiotics amphotericin B that started to be used in 1945. And paromomycin, also another antibiotic. And only in the late 90s that we had a new formulation of amphotericin B, ambisome, which is a liposomal formulation that has revolutionized the treatment because it really improved the efficacy and reduced the toxicity of the previous treatments. And this century, in the 2002, more or less, miltefosine started to be used in visceral leishmaniasis. And since then, we have only combination therapies with the old drugs. No new drugs has been launched since then. So all these drugs, the last ones here, I used mainly against visceral leishmaniasis, liposomal amphotericin B and miltefosine and paromomycin. And with cutaneous leishmaniasis, we still use these first ones here. So the situation is really very bad. And besides the toxicity, these drugs are, with the exception of miltefosine, they are parenterally injected and with multiple doses that causes all these side effects. And for all forms of diseases, no, antimonials, amphotericin B, pentamidine, and they are all used for forms of disease. The amphotericin B, the liposomal formulation, is used mainly for the visceral leishmaniasis, but in some cases, difficult cases, is used for the treatment of mucosal leishmaniasis too. And miltefosine has been recently approved in Brazil. Sorry? No? In 2018, it was approved for the treatment of cutaneous leishmaniasis, but we still don't know how it will be the efficacy still under test. It's used mainly for visceral leishmaniasis. And we don't have any effective topical treatment for the forms of cutaneous leishmaniasis. Although we have on the market, but this paromomycin creams or amphotericin B, they have not been working, especially in the Latin American leishmaniasis. I will concentrate my talk on localized form of cutaneous leishmaniasis. That's what the form of disease that we've been working with more deeply. And why is it so difficult to develop a new drug? That's why I focus it on cutaneous leishmaniasis. And so I've already talked about the systemic toxicity, liver, spleen, heart. Miltefosine is a good oral drug, but it causes teratogenesis, so it cannot be used in women in childbearing age. And WHO and the NDI has always stressed that we urgently need new oral and topical drugs for all forms of leishmaniasis, but especially cutaneous leishmaniasis with the topical drugs. And why is it so difficult to find a new drug for leishmaniasis that still use the old ones? Well, first of all, this is an intracellular parasite, and the drug has to cross two cell membranes. First is the plasma membrane, and then the phagolysosome membrane to reach the parasites, which is in there. And apart from that, we have to think on the route of administration of the drug. That if you want a topical cream, the drug has to cross the stratum corneum, which is a very effective drug barrier to find the infected macrophages deep in the dermis. And if you want an oral drug, you also have the gastrointestinal barriers, which is the very low pH of the stomach and the digestive juices in the gut, and also the mucosa that may sometimes be selective for many drugs, especially the ones with high molecular weight. And in cutaneous leishmaniasis, you have another problem that as the lesion develops and ulcerates, you get a thickened epidermis. So it's not only the stratum corneum here that you have as a barrier, but also the epidermis gets much more, much deeper. And normally here in the ulcer, people think that, well, you have an open ulcer, so it may be easy to treat with the cream because you don't have the stratum corneum here. But that's not the truth, because in the ulcer, you don't have infected macrophages. You have only dead cells and exudative liquid living here. The infected macrophages are below the ulcer border. So you have to have the drug to cross this skin here around the border. That's why it's so difficult to find a cream. Even when you have an early lesion, it is already thickened. So if you use a normal skin as a test, it will not represent what you'll find with the infected skin. And so because the stratum corneum is made of corneocyte, which is made of lipid bilayers, you have these pores here with about 40 nanometers between these lipid blocks. So you need a lipophilic drug to cross the stratum corneum for the first place. And also the drug should not be large. It should be ideally below 500 Daltons so that it can cross the borders or the area around the ulcer. And the drugs that have been mostly tested for the development of topical drug are amphotericin B and paromomycin. And curiously, these are exactly the ones that are the largest anti-leishmanial drugs. As you can see here, 924, 615. And apart from the size, all drugs are not lipophilic. So that makes things even more difficult for topical transdermal crossing. So we have been working here. Here is, sorry, it's the drug discovery cycle where you have the drugs that are selected from collections or natural products, or when you have chemical synthetical drugs, and you do the primary assays on the parasites in vitro. And if they are okay, they go to secondary assays like cytotoxicity, or perhaps to animal studies of efficacy, bioavailability, toxicity. And if you see that the drug lead is good, you can go for structural characterization of protein ligand complex, lead compounds in SAR, and do a design again and synthesize new analogs or new compounds. And this cycle may go on and on until you have a candidate drug that is worth going for a clinical trial. And this process may take years and years, and sometimes just one drug, and it will not pass many of these in vivo assays here after many in vitro testings. And after in vitro, in preclinical tests, you also need a formulation before the drug can go for clinical trial. And after that, it has to be approved by regulatory agencies and manufacturing, and then market. So it's a long way to get the drug to the market, and we haven't got any so far for leishmaniasis after mutafusine. So I will be talking a bit about natural compound drug discovery and formulation. And this is the course of the presentation. Mainly, we have tested many Brazilian plants about 20 years ago. And we ended up with plant substances of the class of chalcones. And we did target identification, nanoformulations for oral and topical delivery. So starting here with the chalcones, the piperidone plant was the source of a very active natural chalcone, which we call the DMC, and because it was very difficult to purify from the plant, and we had synthesized some analogs, and we found this chalcone CH8, which has a nitro group, which was very good, and worked in vivo in mice that were infected with Leishmania amazonensis, and they were treated subcutaneously, intralesionally with 8 injections, and we compared the effect with the antimonium pentostan. We used those 60-fold smaller than pentostan, and even so we found a similar effect in reducing the lesion size. So this chalcone CH8 has been our drug prototype for development. And more recently, we had it synthesized an analog MAP22 that was actually synthesized for the identification of the drug target by Patrick Steele. We had a student in Durham that helped synthesize this. And then another student of ours, Douglas Escrivani, came to join this target identification here. And so Douglas with Patrick in Durham used a very elegant method that Patrick was using, and it's the synthesis of chalcone probe, which has this alkyne group that can bind the rhodamine tag through the click reaction. This is a click reaction here, so that you have your chalcone fluorescent, and you can identify by the fluorescence that it emits. So we showed that when the parasites are treated with this probe and then given the tag, we can assess by confocal microscope that the chalcone is actually inside the parasites. So it can enter the living parasite and be distributed in the cell. It doesn't seem to be focused on the specific place. So we found this intracellular binding, and we found also what was what protein the chalcone was binding to the parasite after lysing the parasites and then incubating with the rhodamine tag. We did the SDS page and identified the band that was more fluorescent, which has the rhodamine tag. And by mass spectrometry, we identified this protein, the cytosolic triphydroxine peroxidase. So we had to confirm that if it was really binding to the protein. So we used the thermal stability and mass spectrometry and also docking. And this was published last year, showing then that the target of the chalcone was this enzyme. And so to confirm, Douglas made this by CRISPR-Cas9, deleted the protein from the parasites, and it was confirmed, and also by PCR. Here, the parasite had a lower survival. And we found that the parasites that had the protein deleted were less killed than the others. So that confirmed that this protein was important for the activity of the chalcone NAT22. This is an important protein for the parasite that is involved in the panothione redox balance, which the parasite uses to detox the oxygen radicals that would kill it. So it's very conserved in trypanosomatids and important not only for survival, but also for infectivity. So that was one of the stages that we passed through. After then identifying the chalcone, identifying that the target, which is important not only to predict the toxicity of the drug, but also the combination with other drugs, and also to help synthesize new active compounds. After that, we went to nano formulations to seeking oral and topical delivery. Leishmaniasis is a very interesting model for nano drugs, because the parasite infects almost only macrophages, which are phagocytic drugs. So the macrophage may eat the nanoparticles and deliver the drug inside the phagolysosome, which is a very effective way to target the infected cells. Of course, non-infected cells also may phagocytose, but we can spare other cells that are healthy around and direct and make the treatment more effective. So the nanoparticles can go particularly to macrophage-rich organs, such as bone marrow, the liver, the spleen, and the skin. And we have the amazon, as I told before. This is one of the first nanoparticulated liposomal formulations, no, with the amphotericin B for many diseases, and it's used also for fungal diseases. But the problem is that it's used for intravenous injections. So we still have the same problem, no? That we need patients to be in hospital. And but there are many advantages of the drug delivery system, the nanoformulations, in comparison to the conventional formulations. They increase the drug stability, increase absorption, increase targeting. This is the good side. And also you may need lower doses, as you see with the Ambisome as compared to amphotericin B. The cost in the end may be less because you need less doses, and also you may reduce the side effects in a very drastic way, also using the Ambisome as an example. And we have many nanoparticles that have been studied for drug delivery. And the dendrimers, colloidal gold, these are metal nanoparticles, and here the lipid nanoparticles, polymeric, cyclodextrin nanocrystals, magnetic nanoparticles. All these, and there are many more. It's a very huge plethora of nanoparticles that you may find in the literature for many diseases, especially cancer, no? And leishmaniasis too. These ones are the biodegradable nanoparticles, so I'm not going to talk about these ones that are not degradable, so they not the approval will be very difficult for these particles in treatment of diseases. So with this one here, we have used the chalcone, which is lipophilic, so it has a very good property to be encapsulated in many different particles. And also, apart from the lipophilicity, the chalcones are a crystalline structure, which is also interesting to make nanocrystals, which is a nanoparticle with the pure drug. You don't need polymer, lipids, or anything else. We have used all these particles here for encapsulation of the chalcone CH8. And we have deposited some patents too. And this one, the dendrimer, it sounded very interesting in vitro, but it showed to be quite toxic for the mice, for the skin. So we discontinued the studies of dendrimers, and also the microneedles, which look very interesting for a skin patch. But the results were very inconsistent, so we didn't go further with these two. With those ones, they were much more promising. And so I'll talk about what we have done recently with nanocrystal production. Very shortly, I'll go through all of them, don't worry. Just to give an idea of what can be done. And in terms of nanocrystal preparation, you can do dry milling or wet milling or both. For the chalcone NAT 22, which you can see the same magnification here for the three forms, and it has very big crystals. And when the powder is submitted to dry milling, you can get smaller crystals that we call micronat. And then you can still you can see some big crystals. You can do wet milling to get in the end the nano crystals here, nanonat we call. And the size distribution of the NAT, the original chalcone is very broad. And around 200, 600 micrometers, whereas the nanonat is around 200 nanometers, which is much smaller, 1,000 times at least smaller. And you can see that the nanonat is quite soluble when you centrifuge and measure the amount of drug in the supernatants. It is much higher in the nanonat than in the NAT 22, confirming the greater solubility after nanomilling. And when tested in mice infected with Leishmania amazonensis by the oral route, we found that this black line is the untreated animals. The blue line is the NAT 22. It has a partial effect only, and the nanocrystals can increase the efficacy, perhaps by increasing the mucosal absorption of the drug, because the chalcone doesn't seem to suffer much from the low pH of the stomach. So it seems to be because of the availability of the drug that can be more absorbed. And here, when we quantify the parasite loads in the ear, infected ear, we find that the nanonat is, as compared with the original crystals, is more effective. And this is a positive control, which was glucantin given by another route, by intralesional route, just for comparison. So this confirms that crystal forms of drugs can be made more effective and even has a promising oral use. The next nanoparticles that we used was the polymeric for topical use. This polymeric nanocapsules is made of a polymer called polyepsilon caprolactone and has an inner oil core where the lipophilic drug will stay very well encapsulated. So the size is about 120 nanometers, which is much larger than the pore of the stratum corneum, which is around 40 nanometers. But even so, you have the hair follicles where the particles can enter and be releasing the drug from there. So they can reach the dermis where the infected macrophages are. So what it did was, sorry, here is just to show that the nanoparticles, when made fluorescent by using fluorescent polymer, you can see that they are inside the macrophages and concentrated here in these small red dots. And this is a sort of lotion that we used to spread on the shaved skin of mice. And after spreading, we used the IVs to measure the fluorescence of the skin through time. And we found that after 3 hours of application, the fluorescence started to go away. And here we confirm that we had a high fluorescence in the beginning, and then it disappeared. It is not just that the fluorescence was quenched, because at the same time that we passed on the mouse skin, we passed on a plastic surface, and the fluorescence was still there. So it indicated that the particles had been entering perhaps the hair follicles and away from detection of the surface. And when the animals were treated, infected animals were treated with these particles, we found really that the number of parasites in the ear was much slower, indicating that the drug had reached the dermis, no? And killed the parasites there. So this was a work that we published in 2019. And so we demonstrated that polymeric nanoparticles, although much larger than the pores of the stratum corneum, can effectively deliver the drug in the dermis to kill the infected, to kill the parasites. We also used polymeric microparticles now, and not nano. It's a bit larger, because this is a depot particle that we want to be releasing the drug gradually. The idea is to reduce drastically the number of doses that you need for intralesional injections that use with the antimonials, because the antimonials are very hydrophilic, so you have to keep repeating the injections because it's absorbed to the circulation. So we want just one single dose to treat cutaneous leishmaniasis. Sorry. So what we expect is that after injection, the drug is released continuously, while the biodegradable polymer is hydrolyzed in acid lactic, which is innocuous, and the drug is released. So we propose to inject around the lesions, no? These microparticles that will be releasing the drug for weeks. And it's a mixture of nanoparticles and microparticles, so that the nanoparticles is first phagocytosed by the macrophages, whereas the larger microparticles will be releasing for weeks by the depot that's formed under the skin. And we have prepared by solvent emulsion and solvent evaporation process these particles. And you can see here in electron microscopy on the particles, how different the chalcone looks. Here is the fibers that looks like needles is transformed to much smaller, you know, spheres, because when you use the chloromethane, the chalcone dissolves and is encapsulated inside the small microparticles. And here is the size, it's around 6 micrometers, whereas each needle here may have 500 micrometers. No? So it's much reduced. And this is the schedule of the treatment. The animals received either 1 dose alone or 3 doses just to maximize. And we used the glucantine as a positive control. So you can see the ears, how it looked, the untreated ones, and here with 1 single dose. And 3 doses here, it looks like it completely returned to normal appearance, no? The ears. And here the parasites in the infected ear that is reduced even with 1 dose alone. Of course, 3 doses was more effective, but we want to stick to 1 dose. So we still continue to improve the method to try to reduce the treatment to one dose alone. So this is published in 2018. We improved the encapsulation rate by using a spray drying technique, which increased about 10 times the amount of drug inside the polymeric microparticles. And this is one method that we may pursue, one of the 2 methods actually, because this is spray drying is a method that can be used by the industry to produce the medicine, whereas the bench method that we used previously, it's not scalable. So we plan to stick to spray drying, and also another method that we are about to compare which one will work better, which is the microfluidics. And this microfluidics is a new method of production of nanoparticles that is scalable, from bench to industry, which is interesting. And it is being used also as an organ on a chip or body on a chip. And we are now with some partners at the university producing with the 3D printer these devices here so that we can be self-sufficient. They are quite expensive to buy, and I think we are going in the right way. The advantages of this microfluidics in comparison with the conventional method is that with conventional method you have a spread by size of the particle with a broad range of size, whereas the microfluidics you have a very narrow dispersion, no? So you can have a more controlled production. Oops, sorry. So just to finalize, we are now carry on that way. And coming back to this scheme, I'll talk about the compound collections, no? That Paul Denny has some time ago got from GSK and tested many compounds that were at GSK. And he identified the clemastine fumarate that together with Patrick Steele that synthesized some derivatives. And we have published together 2 years ago, this paper, where Paul used the phosphoryl-ceramide synthase inhibition assay. He has a platform to test new drugs there, and he identified the clemastine fumarate, which is a drug that is on the market for the treatment of allergic diseases. So it's a repositioning strategy. Here the clemastine fumarate, no? And the inhibition of IPC. We demonstrated that clemastine is very active against many species of Leishmania, of cutaneous, that cause cutaneous and visceral leishmaniasis. And the range of 0.027 micromolar. So they are very active, the clemastine fumarate, against promastigotes and amastigotes. And when tested in vivo by the different oral, routes, we tested oral, intraperitoneal, and intralesional routes, and we found that when given intraperitoneally, clemastine is very effective in reducing the lesion growth, but it was not the effect by the oral route was not so good, just a partial reduction, which was very interesting because the clemastine is given by the oral route to humans, but not didn't work that way in mice. And the intralesional also was quite good, but we still want to stick to the oral administration because that's what the drug is used in humans, no? So we have used the two approaches to improve the clemastine uptake by the intestine. One is by using the beta-cyclodextrin, which is a sugar polymer that you can, it's well used by the industry and by some pharmaceutical production too, to encapsulate lipophilic drugs to make it soluble in water. So that's the one way to make it more soluble. Perhaps for mice it would work better in that way, in that presentation. And another presentation was in polymeric nanoparticles that was encapsulated PLGA nanoparticle, which is about 200 nanometers in size. And these were given to mice daily for 15 days. And then we measured the parasite loads in the infected ears. And we found that the beta-cyclodextrin was not very good, not so good, it wasn't different statistically, but the nanoparticles seem to have improved the, I don't know if the absorption or if it was stuck to the hair follicle too, like I had shown before. But in this way, sorry, okay, I was mixing up. This is by the oral route, so it is just by the absorption through the gut mucosa, not forget about the skin. So the nanoparticles seem to have been absorbed much better than the free form of clemastine or the cyclodextrin. We are now repeating this because we had to do just a partial treatment 15 days. We are going to try to maximize for 30 days of treatment and see if we can get a good result with the oral treatment with clemastine fumarate. Well, this is the end of my presentation here. I went through this just to have an idea of what we can do from screening to delivery. And we are at this stage now in another formulation that we have with the amphotericin B, just to make the approval easier, because if we use chalcone in humans, it will have a very long path to go because it's a new drug. So we want to open this path with a known drug, which is amphotericin, and then maybe try with another combination of drugs, chalcone or any other drug that looks promising. That's for the depot, subcutaneous depot formulation. That is, has a clinical trial that will come up soon. We are at the moment doing the good manufacturing practice production of the formulation. And I want to thank all my collaborators from the UFRJ. This is our students, Camila, Wallace, Douglas, Felipe, Maria Paula, and from the COPI, which is an Institute of Engineering at UFRJ, Ariane and Thiago, from Federal University of Rio Grande do Sul in Brazil, Adriana Polman and Sylvia, with the first polymeric nanoparticles used. And in Durham, my colleague, dear colleagues, Patrick, Paul, Emka, and Stephanie. Rebecca Charlton was our PhD student with Patrick and Paul. And in France, I have also Marinese Hair, and all the agencies that allowed this work to be completed. And thank you all for your patience, and forgive my voice. I'm really a bit rough today. Sorry about that. Thank you very much.
Thank you so much, Bartira, for such a nice presentation. I have a couple of questions for you. One of the question is from Solomon, and he's asking about that. Kindly elaborate the target identification protocol you use in your study.
I'm sorry, I'm trying to close here. Now I can see. Sorry, what was the question?
The question is that kindly elaborate the target identification protocol you used during your study.
For the target identification, what was the, sorry, Nahid, I couldn't understand your question.
Bartira, I think he's asking about the target identification. I mean, what protocol you used to identify that specific target, and so kindly elaborate that. Yeah.
Oh, you want, but is there any specific question about the protocol? This is a protocol that Douglas did with Patrick in Durham. So they used synthesized a chalcone probe with that alkyne group that when you give to the parasite, it will enter the parasite, and then you give the fluorescent tag so that you can you can measure where the fluorescence is. You want to know specifically how I treated how the parasites were treated?
I think because the question is not very clear, but I would suggest that I think this work has already been published, so we can share the reference of that paper. So
yeah, we have all the protocol described there with details. If you want, you can write to me and I can give you the paper. Thank you very much for your question. Sorry if I didn't answer correctly.
Bartira, one more question from my side. What do you think about the use of multifocine for the treatment of cutaneous leishmaniasis?
Is that Nahid who is asking?
No, it's me who is asking.
It is true.
Yeah.
I thought Nahid was wanting to ask something. Sorry about So multifocine for the treatment of cutaneous leishmaniasis. Multifocine has been approved in Brazil for treatment of visceral leishmaniasis in dogs. And that was a big problem because now we cannot use that in humans anymore. In human visceral leishmaniasis, because we will have Leishmania infantum that may develop some resistance to multifocine around. So it was approved only for cutaneous leishmaniasis, but we still lack some more broad results. We have some clinical trials. Some shows that it works, some shows that it doesn't. So you see now when the doctors start using it more openly in the clinics, maybe we have some results in some earlier times, perhaps.
Yeah. I asked you this question because I was going through the literature and I found a paper that was published by a team who was working in northern areas of Pakistan. It was about 3 years back, and they published a study in which they claim that they used that multifocine for the cutaneous leishmaniasis and it was effective. So I will share that paper with you also, because after that we are thinking about that we can use that, but I think but little bit with modifications, because multifocine is not approved yet in Pakistan. Yeah.
Yes, well, it may not be effective in Latin American leishmaniasis, but it may be effective in the old world. So there are very different species. You have, in Brazil, the cutaneous leishmaniasis is not self-resolving as it is in the Middle East and in the old world. So the parasites may also behave differently. So I think it should be tested according to the species.
Bartira, we have two questions and both of them are actually related with the selection of chalcones. One of our participants is asking that if only chalcones compounds should be used as a base to fight against leishmanial disease, number one. And number two, the other one is asking that what was the rationale on selecting chalcones for your study?
The first question is if the chalcone, sorry.
Yeah, they are asking about that. Is the chalcone is the only choice to fight against leishmaniasis?
The only price? Well, it is our prototype drug. Many people have confirmed that chalcones have good anti-leishmanial activity against many species of parasites, not only the cutaneous, but also visceral leishmaniasis. And so there was the first chalcone that was discovered, which was the liquid chalcone by some people in Denmark, but he was from Sudan, but was working in Denmark. And that was the first description. After that, we published, and then many other groups have confirmed the activity of chalcones. It had, in our case, the nitro group seems to confer a higher activity. It's important for the activity. So the second question was?
The second question was also related to that what was the rationale of selecting chalcone for your study?
Why did
why you select chalcone for your study?
Well, for the nano formulation, we selected because, first of all, because it was very active against the parasite. It was difficult to work in vitro because it was very poorly hydrophilic. And exactly because of that, of the lipophilicity, it looked a very good drug to work with the encapsulation in nanoparticles, because if you need to use organic solvent to solubilize the chalcone or the drug, which is compatible to the ones you use for the polymer solubilization. And the polymer is not soluble in water, so you need to have a lipophilic drug. Of course, for liposomes, you have an aqueous core that you could use a hydrophilic drug there. And we have used also liposomes, but in that case, the drug will remain in the bilipid layer, no? Of the liposomes, not in the aqueous core. So it can work for polymeric and lipid nanoparticles. That's why we chose this lipophilic drug. And also because we can compare, no, with which nanosystem would work better with using the same drug.
So with this, thank you so much, Bartira, for your very nice presentation. And we all wish for you and your husband for a speedy recovery from the infection. And we will share with you the all chats and the link of your lecture for your record. And for the participants, that let me inform you that the lecture will be available on our YouTube channel also. So bye-bye, Bartira.
Thank you very much, you all, for your attention. Bye-bye.